So I've said this a few times in class. The basics of this process are that DNA, inside your DNA exists this code that specifies all of the proteins in your body. The problem with this is that DNA lives inside of the nucleus of your cells. Remember, we are all eukaryotes, and so we have to have these nuclei, which is the plural of nucleus. We have to have these nuclei, which is where all of our DNA is going to live. But the problem is that proteins are not made in the nucleus. They're made in the cytoplasm. The cytoplasm is just the stuff inside your cell that isn't an organelle. All right, so you have DNA, you have the instructions living in one place, and you have the location that those instructions are needed living somewhere else. And the solution to this is transcription. And what transcription basically means is you can think of the DNA like the library, or you can think of it as like a really important reference textbook that you don't want to get messed up. But you need to get stuff out of it. You need to get information from it. And the way that you get information from it is to go into the library, take a pen, take a pencil, copy down the information that you need. The library would be kind of like the nucleus here. The reference text that you're copying out of would be like your DNA. So you go in, you copy down the information that you need, and you take it with you outside of the library. The copy that you made of the DNA would be your transcripts, or that would be the process of transcription, copying down information you need and taking it out of the nucleus. So, like I was saying, the cell is making a cheap copy of DNA in this less permanent form, which is RNA. We've learned a little bit about RNA during the semester so far, but RNA is able to leave the nucleus. Okay? And the process of copying DNA into RNA is called transcription. So just to make sure we're all on the same page, inside your nucleus you have DNA. DNA is the instructions for protein. Protein is not made inside the nucleus, it's made outside the nucleus. But you can't just have your DNA leave the nucleus. That wouldn't make sense. It would be cumbersome, it would be dangerous for your DNA. So what do? You go into the nucleus, you make a copy of what you need to know, of how do you make this protein. That copy can then exit the nucleus, because if something happens to an RNA transcript, it's not going to be the end of the world. Okay? If something happens to your DNA, it's going to be bad for your cell. Okay? And that process of copying DNA into RNA is called transcription. So in a moment, we're going to talk about how our nucleotides translated from how are nucleotides translated to amino acids. Okay, and proteins, like I was saying in the last video, are assembled from amino acids on ribosomes. There are long chains of these amino acids, and the correct amino acid is added at the correct time using information from the RNA message from the nucleus. Like I was saying, you're taking those instructions, you're bringing them outside the nucleus, and you're using them to construct a protein. Okay? The process of constructing one of those proteins is called translation. And we're going to talk about that in a minute here in the next video. But transcription is getting the instructions from the DNA into RNA, which is this cheap, it's scratch paper. Basically, you can think of it like that. Getting that from the library and actually building something with it. Translation is the process of that building. So here you have your DNA, and you can see it's starting to partially unwind, and you're getting your RNA copy of it. This is transcription. The RNA copy of it then leaves the nucleus and is translated into protein by a ribosome. And I want to point out here that the copy that you make from your DNA is called mRNA. mRNA stands for messenger RNA. So DNA and RNA are slightly different molecules. RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. As we kind of learned about earlier, RNA is slightly different. It has an A, G, C, and a U, whereas DNA has A, G, C, and T. And RNA is just single-stranded, whereas DNA is double-stranded. RNA is a single helix, DNA is a double helix. So this is showing you some of the chemical details of what's going on here. I'm not interested in you knowing that RNA has an extra oxygen on this carbon of this pentose sugar or this ribose sugar. Not interested in that. Okay, I am interested in you knowing that they are chemically different somehow. And the real reason is that there's an oxygen here on ribonucleic acid, and there's no oxygen here on deoxyribonucleic acid. But like I said, it is sufficient for you to know that they are chemically different molecules. The other thing you should know is that RNA has uracil, whereas DNA has thymine. Okay, everywhere where you will only see uracil in an RNA strand, you will only see thymine in a DNA strand, and so that's a great way to be able to identify them. So the third thing you should know is that RNA is a single helix, whereas DNA is a double helix. So 3 things to know. DNA and RNA are chemically different. DNA has thymine, whereas RNA has uracil. And DNA is a double helix, while RNA is a single helix. 3 things to know. So there are 3 types of RNA. There's mRNA, which is going to be the transcript from the DNA. mRNA is going to be the copy of that DNA that you're actually going to use as instructions to build a protein. rRNA is the ribosome. In essence, there's some other enzymes in there, but rRNA is the actual ribosome that you're going to use to construct your protein. And tRNA is involved in bringing in the appropriate amino acid at the appropriate time in order to build up that polypeptide, in order to build up that protein. So mRNA is the transcript. It's the instructions for building. rRNA is the ribosome. It actually does the building. And tRNA is what brings in the amino acids to join up and form that big protein. So here's just a picture of them. It shows you kind of where they live. This is your transcript here. This is in the nucleus. It starts in the nucleus and then it migrates out to the ribosomes in the cytoplasm, carries the instructions for protein. Here's your tRNA. This is only in the cytoplasm, and this is actually bringing in amino acids at the right time. It's following the instructions of the mRNA and the ribosome, and it's bringing in the right amino acids. And then rRNA is kind of like the construction site. Okay, it is the structural component of the actual ribosomes, and it's the spot where you're actually going to be building your polypeptide. So again, mRNA is in the nucleus, and then it leaves to the cytoplasm, which is again just the part of the cell that it's just the inside of the cell. Okay, the tRNA also in the cytoplasm, so that's not in the nucleus, it's just inside the cell, and then rRNA in the cytoplasm, just inside the cell. So base pairing, as you can see in figure 14.4, is the mechanism that is used to copy DNA and transcription. Okay, but here we are turning it into RNA and not into DNA. So if you remember the process of DNA replication, the DNA is going to split open, and then you're going to add base pairs to either side of that DNA strand. Here you're doing something similar, except for you're just building RNA. You're just building it with RNA, and then that RNA is going to detach from that piece of DNA, and it's going to leave and go build a protein. Okay, so DNA is unwound and used as a template to match complementary bases to one another. C's are matched to G's, and A's are matched to U's. So you'll remember C's get paired to G's, A's get paired to T's if you're talking about DNA, but in RNA, A's are matched to U's, and you're going to use those to make a new daughter strand. And I'll talk about this in lab a little bit just to make sure that everything is super clear. If you have questions, please ask me during lab. This can be a pretty complicated process, especially if you're just trying to listen here. So there is an enzyme that is critical to building your RNA from your DNA. So remember what just happened is we have our strand of DNA, and we unwound it just like we did in replication. Okay, and if we were replicating, we would use DNA polymerase. But in this case, we're going to use RNA polymerase because we're building an RNA molecule, not a DNA molecule. Okay, RNA polymerase, we're going to use RNA polymerase to build an RNA molecule. That is an enzyme that I would like you to know. Okay, the two nucleotides are linked together by the same enzyme to make a single strand of RNA called a transcript from one strand of the double helix. So what this means is double helix is double-stranded, obviously, and you're going to unwind it, and you're only going to be looking at one side of the double helix in order to make your strand of RNA. So here we go. Here you have this piece of DNA that has been unwound here by, you'll probably remember, DNA helicase. And then you're having your RNA come in here, and you're building this RNA strand from one side of this. All right, so this is being put together by this enzyme called RNA polymerase. And you can see it is base pairing. These complementary base pairs are going on here. C's are matching with G's, G's are matching with C's, U's are matching with A's instead of T's. Okay, U's match with A's instead of T's. And then what's going to happen is this RNA molecule is going to actually break off of here, exit the nucleus, go to the cytoplasm, and be used as instructions to build protein. So like I just said, if this was just a replication process, this would not leave. It would stay here, and your DNA would just be copied. And then ultimately, your DNA, after it's been unwound so that you can build this RNA, is going to get stitched back together. So the final step here is mRNA processing. So you can't just have an mRNA come off of the DNA and be ready to go. It has to be edited further. Parts of it are cut out, which are called introns, or intervening sequences. Okay? Introns need to be cut out of that piece of RNA. The remaining pieces are called exons, which are joined together to make the finished product. And you should probably be reading about junk DNA. There's a little section in it about that in the homework as well. So the basic process is you unwind the DNA, you make an RNA transcript, you edit that RNA transcript exits the nucleus, meets up with a ribosome, which consists of rRNA, and then tRNAs are used to bring in amino acids. So this is the process of editing out those introns. I'm not worried about you knowing like the biochemistry of this. I just want you to see kind of how it works. So here you have exon, intron, exon, intron, exon, and you have an enzyme that kind of forms this loop and cuts these pieces out, and then you wind up with just exon, exon, exon. And those sequences are gone. And there's actually a pretty good reason why you might do that. Maybe we can talk about why you might do something like this in class later. So I'm going to talk about the importance of the genetic code and what a codon is in the next video.